started, everyone. Welcome to another session of our evolution seminar series. Today, we're going to get liminalogical as we are joined by Trina McMahon from the departments of bacteriology and civil environmental engineering here at UW-Madison, who's come to discuss some really interesting ecology and evolution in a lake that's very close to home. So please join me in welcoming Professor McMahon.
Good. Sure that this is in the right place. Hi, thanks for coming out during a time where it's really easy to just like sit and doom scroll. I'm hoping I can distract you a little bit, at least for an hour, and maybe get you excited about science, or hopefully sustain your excitement about science. Yes, my name is Trina, and I'm sort of an odd bird. I am an engineer by training, so I came into UW-Madison through the civil and environmental engineering department, but I always did microbiology in wastewater treatment since my master's degree, actually. And then when I got to Madison, I got even more into microbiology, studying the lakes. And I got my appointment split in half. So now I'm 50% in bacteriology and 50% in civil engineering. So that explains that strange combination. So I'm going to talk a lot today about Lake Mendota. And I think many of you know what that is, but I'll give you a brief introduction on why we study lakes more broadly. Freshwater lakes, we like to say, are where all of the biogeochemical cycling sort of comes to a focus in the landscape. Yes, there's a lot of carbon and nutrient cycling in soil, but like a lot of that eventually makes its way into freshwater lakes, and there's just a lot of microbial activity that's really important for, you know, sustaining that ecosystem. They're also really good models because they're like islands, right? So they are somewhat constrained in the forces that can act on the microbes in them, but they also are wild enough to be a bit more relevant and interesting compared to some of the other systems that I study. And of course, we all value them, right? People in Madison especially really value their lakes, well, in all of Wisconsin, because they provide recreational resources and fish and all the things that humans care about. So we study them for lots of different reasons. I'm going to focus today mainly on the microbes and from a more kind of basic science perspective. So you can't quite see the cyanobacterial bloom here in Lake Mendota, but you'll recognize the overall shape. So Lake Mendota is experiencing these multiple drivers that are impacting both the water quality and the microbes, as well as things like the fish and all the other stuff that I mentioned. And those range from at different time scales, right? So you have the gradual long-term change that's coming from our climate change. We have these flashy changes in precipitation patterns where we're getting more floods and more droughts, kind of like that whiplash in the weather. We've had 2 invasive species that have invaded the lake in the past 20 years. And I'm not going to talk much about them today, but we have a really solid area of research on that as well. And then we have land use change, because as you can see, and you may know, there's a lot of urban, suburban development around Lake Mendota, but there's also a lot of agriculture, at least to the north of the lake. So up here, which I'm not showing, and that contributes nutrients into the lake, like nitrogen and phosphorus, that fuel all that green scummy stuff that you see in the summer, which are the cyanobacteria. Also, which aren't going to feature too prominently today, but hopefully I'll have some time to talk about them at the end. So one of the foundational principles that we have in our work, we borrow from what's called the long-term ecological research site, or there's actually a network for it. And it's grounded in this concept of the invisible present. And so John Magnuson was the first PI of our local LTER, and he wrote a really famous paper where he introduced this concept using Lake Mendota as the example. So in this case, we're showing on the y-axis the duration of ice cover, right? So in 1983, which is when this LTER started, you could see, you know, ice cover was about 50 days. And so, you know, if somebody asked you, so about how long does ice cover last on Lake Mendota? You say 50 days, right? So the problem is that if you start going back in time and a little bit forward in time, you can see that that was actually quite a low point in our the dynamics or variation in the amount of ice cover. And so it might be more, you know, like 90 days. So if you keep going, you know, you can see the amount of variation, and then, you know, you could maybe draw a line through that to come up with some average that you could guess it might be in a particular year. And you keep going. Now you can see a long-term, downward trend, and maybe also differences in variability over certain timescales. And when you put it up till about the present, you can see this sort of precipitous drop in the long-term change that happens around 1975, which is when a lot of climate change signals really became really obvious in the data set, these long-term data sets. So we're really fortunate to be partnered with the LTER. The NTL stands for North Temperate Lakes. So we call ourselves the North Temperate Lakes Microbial Observatory, and we're kind of like an appendage of the main LTER. So we work really closely with the folks over there. So I had a graduate student, her name is Robin Rower, I'll talk more about her later, who was really interested in this long-term time series of microbial. We have both 16S ribosomal RNA gene sequencing, which I'm not going to talk about, but she did some work on that, and that she published in 2023. And then the second part of her PhD, which then also became her postdoc project, was to look at the genomes of organisms across this time series. So to help you interpret this plot, I have the month or time of year along the x-axis, and then on the y-axis are years, right? So starting, well, now it's 25 years ago, but at the time she processed all the samples, it was sort of early 2019. And then the color coding is the season that we define by various stuff that happens in the lake, or in the case of ice that, everybody knows what ice on and off are. But we're not going to talk too much about that seasonality. But each dot is a sample that someone collected going back all the way to, my very first graduate student. So we also have a very strong culture in my lab of the pay it forward. So everybody, has to contribute to some kind of long-term time series where they collect samples for, the graduate student that I retire with, right? So we'll see how long we can keep that going. And you can see there's certain years we had really dense sampling. That's actually the year that Robin joined the lab. She really wanted to double down on that. But we have pretty good coverage across at least the open water season, and especially in the sort of June, July, August, when we have a lot of undergrads helping us for the summer. So she wanted to ask, okay, we've got the bacteria in the lake that we know are changing because of ecological drivers. So interactions with other organisms, environmental factors like temperature or nutrient concentration, and all those things that you might initially think would be driving the ecology. And there's lots of papers published about that. Both, we've done a lot on Lake Mendota, but then other people, that's kind of like the thing in microbial ecology, is to look at those factors, right? And what Robin was really interested in doing, though, was diving into the interaction between like ecological forces, if you want to call them that, and then evolution of the most abundant organisms in the lake. So to do that, we use something called genome-resolved metagenomics. And by now, this is a fairly mature approach. There are a lot of workflows and bioinformatics tools to process big metagenomic data. And it's still kind of hard to do like big projects like we undertook. We had a lot of support from the Joint Genome Institute to do this. But we were then able to process a lot of those really big data sets and generate what we call metagenome assembled genomes, or MAGs. So really briefly, that workflow says you take a sample, you extract the genomic DNA, just like you would from an isolate, but you're doing it from a community of microbes. You then put it into a sequencing machine. In this case, we were using Illumina, so we get short reads. So this is your short read sequences from all of the microbes that were in your sample. Then you munge it through a bunch of computer stuff, and what you end up with is a genome, hopefully. That genome of a particular microbe, you can then study using the mapped metagenomic reads, which I'm going to talk about because this is a little bit hard to comprehend. So I'm going to walk through some of that because it's really fundamental to the rest of the data that I'm going to show. So bottom line is that you can go to a water sample, you take a 500 milliliters of water, and what you get back is like 10,000 genomes, right? If you do all of this fancy bioinformatics. So when Robin did this, She had 471 metagenomes. Some of them were duplicates of time points to check as a control. These are the stats on the amount of compute time that she used. She did this with the University Texas Austin because they have a really awesome computing facility, and that's where she was working with her postdoc advisor. And so she got 85,000, about 86,000 sort of the raw collection of genomes that we get out of it with varying quality, right? So sometimes the genomes look very complete, almost like an isolate genome. Sometimes they're very fragmented because they, for whatever reason, didn't assemble or emerge from that the process that we use to collect the genomes. So there are about 30,000 that had a medium quality that we would consider working with going ahead. And then if you dereplicate that, so like the interesting thing that we find is that if you have 471 samples, there are genomes that you see over and over and over again, right? So there are some organisms that are in the lake all the time, right? Some that only come and go, a few samples. But when you dereplicate all of that, so you only have one representative of each organism, you get about 2,900 or so, right? And so that's, for those of you who think about this stuff, that's clustering at a 96% average nucleotide identity. And this just shows the yield of that, if you want to think about it that way, by phylum, and their relative abundance across the entire data set, right? So across all samples. And so you can see that there's certain phyla that are overrepresented by far. And we already knew that from 16S ribosomal RNA sequencing, which if you don't know what that is just a way that we can count the species without accessing their genomes. And so we got, of course, what you would expect to find, cyanobacteria. Those are the ones causing the nasty blooms. And we found some of the weird candidate phyla, if you've heard of them, which are sort of pretty recently discovered phyla of bacteria that, have been found in lots of different environments. And actually, we didn't know we're in Lake Mendota because they don't match our 16S primers, which is a story for another day. But so we have this big collection of genomes. And if you, again, were like a freshwater aficionado, you might recognize all these acronyms and different, I guess some of them are families, some of them are genera. So you don't need to worry about that. This is the rank abundance curve for, I can't remember how many genomes I did this with, but the blue shows the ones that were detected in all of the samples, right? So we've collected a lot of the most abundant ones, or persistent ones, I guess we should say. So I'm going to do a little vignette on one group of organisms, and that is it's actually the second most abundant right here. The topmost abundant will appear a little bit, but probably the more interesting data that we found, at least so far, is in that second most abundant one. And actually, I should say that, how do I describe this? So this is basically like a genus, right? So there's a single genus, and there's really only one species. This has maybe 5 species. So there's also diversity embedded in here. And one of the reasons why we picked this second one is because it only has one species. So to get our brain around that diversity, we picked the easiest one, because where are you going to start, right? So that's the genus Nanopelagicus, and this shows a phylogenetic tree constructed with genomes. There are some isolates of the genus Planktophila, which is in the same family, but there are no cultures of the Nanopelagicus. This is the group. I just put a box around it, but you can see a lot of numbers. These are single cell genomes that we collected from Lake Mendota, and then some enrichment cultures have genomes from them. But basically, we're looking at that phylogenetic group, if you care. And again, these are found in lakes all around the world. We call them ubiquitous or cosmopolitan because they are everywhere. We know a little bit about what they do, but not a whole lot. So this is one of the more abundant members of that genus. And I'm showing the year, which has all the different samples, and then the relative abundance of that genus in the sample, right? So there is variability, of course, but you can also notice that these percentages, oh, is that? I wonder if that's relative abundance rather than percentage. Might be relative abundance instead of percentage. I'd have to look back because they're really abundant. So that seems really low if it's percentage. And I'll blame it on Robin. So this is not from her paper. This is from another talk that she gave. So she may have made a mistake, but you can see that it's always there, right? Now, the way that I did this was by taking a reference genome from that genus. So really we had only a couple of reference genomes from that genus. And going back to this workflow of how we deal with it computationally, we can take the genomes that we get and then use the metagenomic reads that we use to generate that genome and map them back. So if you don't know what mapping is, it's like BLAST, right? So basically you throw little spaghetti pieces against the wall and ask how many of them stick based on, well, in that case, how sticky the spaghetti is, but it's basically how identical or how similar those reads are to the reference genome, right? And this is also, I feel like, always hard to conceptualize. So what I'm showing here is a chunk of one of those genomes, and these black rectangles are the reads, right? So they're little short pieces of DNA. And this piece of DNA, this contig, has about 70-fold coverage in the lake. So if you went to any particular base in the genome and you slice down through here, on average, there will be 70 reads that mapped against it. So imagine if you do culture-based comparative genomics. So this is like having 70 isolates, right? To be able to look at the genome level variation. 70 isolates from the same population, right? Because we pulled it all out of the lake in one sample out of the lake, right? So we can quantify their abundance using the number of reads or the coverage of that particular genome. And so that's how we get these data, right? So mapped reads onto a reference genome. Robin, I mean, this was something that I've been working on maybe for like 12 years, because I had a former student who was trying to get there as well, but we didn't have the data that was needed to really get into it. And it's this question of like, okay, we have a genome and we have the spaghetti that map to it, but like, is that a species? Is the genome a species? Is it we call them population genomes because they're really assembling together from populations, not from isolates. And then, what are the reads, right? So the way we've started thinking about it is, okay, you have this community that you get a collection of genomes from. And in this case, we have defined our mags, like this, right? So there's mags that are not complete, but we have some confidence that all of the contigs that we got fit into that genome, right? And if anybody cares later, I can talk about the bioinformatics, but just think of it as a genome at this point. Now you can start grouping those genomes based on their percent identity to one another, sort of how closely related they are, and call it a strain, right? Most people think of strains as a clonal like isolate, but we know that even when you grow from a colony into a tube, that you can get changes or mutations in the genome that, might eventually create multiple strains, right? If you've heard about the E. coli long-term evolution experiment, that's exactly what they have been doing for, what is it, like 30 years or something, 40? I don't remember. But then you can group them further into something that maybe we could call a subspecies, and eventually you could group to the level of species and on the way up in terms of sort of the way that we organize biology, right? So we started wondering, okay, well, we need at least an operational definition because we can argue about species concepts, but that's like and definitions, but that's super controversial in microbiology for really dumb reasons. So I'm not going to talk about that. But basically, we're like, okay, we're going to say a genome is like this entity that allows us to look at the population, right? It's basically just the bait to do that mapping, right? So you can't get this collection of reads without having the genome. And so we treat that as, sometimes we use the word species if, the reviewers will let us get away with that, but oftentimes we're just talking about it as a MAG, right? A metagenome assembled genome, because we don't know what else to call it. But then the reads allow us to look kind of at the level of strain, we would say. That's sort of our current concept. And that has to do with the, really trivial things like what's the cutoff for the spaghetti, right? Like how done does the spaghetti have to be for it to stick to the wall? And what is that number, right? Is it 97% identical? Is it 95? And there's no guidance in sort of the field of metagenomics on what to use for that. So we generally use 97% to define, I guess, the population, which is multiple strains present all at the same time in the lake. Okay, so what that looks like when you start zooming into those reads, right? We can get abundance using the reads, but then we can also get variation. And it's a little bit hard to see here, but maybe in the back, but maybe you can see there's like this column of blue. That is a single nucleotide variant, we call it. Some people talk about SNPs, right? But we call them variants. I think it's because it's in the at the population level. But mostly this is pretty, homogeneous, not a lot of SNVs in it. So this would be a region of the genome that in that population was pretty, what you would say, I don't know if you call it highly conserved, fixed, like whatever term you want to use from population genetics, which like starts to get a little hard when you're thinking about it this way, with reads instead of organisms, isolates. But so this would be an example of high percent identity, and this would be lower, right? So in this genome, you can still get the spaghetti to stick, but there's, pieces that are kind of hanging off the wall, right? Because they don't match perfectly. So we can quantify how much of this is in the population across the entire genome. And these are from the same contig. So there's variation across the genome, which we kind of just don't think about because it starts to get to be too much. So we think about it at the average sort of across the genome level. So this is a kind of plot that you could get when you do this analysis. This is kind of an old way to look at it back when we only had like one metagenome and one genome. But I'll walk you through it because I think it also helps conceptualize what the rest of my plots are going to look like. So this is again a genome chunk. And now we have each point being a read. And the y-axis is the percent identity of that match. So like how, I guess 1 minus the number of mismatches when the spaghetti sticks to the wall. And so what people have noticed is that there's this kind of like region in the percent identity space that is consistently like there's not very many reads there, right? And so this was something people noticed in the very early days of metagenomics and said, this should be like our species boundary. Like anything that's more similar to the reference genome would be a member of the same species, and anything that's less similar would be another species, right? So people argued about that for a while. But I think the way I've started to remind myself every time I get stuck in that hole of arguing about it, I look at it this way. So now I've turned that plot on the side. And I have percent identity on the x-axis, and this is just like a, if you want to think about it, like a histogram, right? So it's density of the number of reads that are mapped at that percent identity. So these are the really, really high percent identity. So we call that, or we used to call it a sequence discrete population. And we don't use that term as much anymore, but because we start, like, is that a population, right? So we think about, is that a population? So now we just call it population, but there's this discontinuity where there's just not very many reads in the population that match your reference genome at that level of percent identity. Nobody has a mechanistic explanation for why we see this. So I don't want you to think that we have this figured out. Some people argue, vociferously that there's many species inside of here, right? But it depends on what you define species and all those things. So we're going to operate with this slice, right? So just the stuff that's greater than about 97%. So that's what this is, right? So the spaghetti could only stick to this reference genome if it was 97% or greater. Again, kind of an arbitrary cutoff. All right. So I mentioned that you can quantify the amount of the extent to which the population matches the reference genome. And you can use that using metrics that have been developed for population genetics. And so on the y-axis now I'm showing what is called pi. It's a measure of nucleotide diversity We have software packages that just calculate this based on all those mapped reads that I showed you, and this is averaged across the whole genome. So now I'm showing the year, and there are samples across there, and this is the extent of population diversity in that sample. So I kind of, the way I think about it, and you know, folks who do real evolution stuff can laugh at me for this, but I think of like, if you have a species in the lake, the diversity within that species, I think of it as a cloud around that species, and it can kind of, if it expands, you know, with mechanism unspecified here, right? So there could be more diversity, less diversity, right? And so higher pi means more diversity, lower pi, more clonal. I don't know, are you even allowed to say that? But more as if it's a homogeneous population. And so the first thing that jumped out was like, okay, what happened here, right? So you've got your time series is going up and down. You know, we could talk about how you can interpret the increase and decrease of nucleotide diversity, but we do see this pretty sharp, almost what a limnologist would call a regime shift. So a new stable state of level of diversity within that population. And so we scratched our head about this a lot and asked, well, like what could be causing this? So the population became more diverse. What does that mean? Could we have had immigration of a new strain that now has taken over, but, you know, or replaced some of the strains that were there previously? And so we looked at just counting up the number of new SNVs, so the different colored columns that I showed you in the mapped reads, how many new ones appeared in each sample. And so, of course, at the very beginning where you're sort of inventorying all those SNVs, you know, it's high, but then as you go through time, there's no new ones that have appeared in the lake, or very few anyway. So probably not like an invasive strain of this organism. And so we kind of eliminated that as a possibility. So now we're thinking that it's more of a shift in the relative composition of the strains, which maybe that's ecology and not evolution. But if we're defining them as strains within a species, is it? But we don't know why they changed. Is it because some got selected for and others didn't, which would be, is that ecology, right? So like, this is where we came up with this idea, okay, we're working where in this gray zone where we don't have the vocabulary really to even talk about it because we can't define what the species is. What it always comes down to. So, you know, because we're also limnologists, we wanted to understand, okay, like what were the forces acting on these populations that might have caused that change? And this is where that invisible present concept comes in, because we have this 20-year time series and we just, oh, something happened when, you know, what day did it happen on? And then maybe we can look at what happened around that time and make some inferences to what actually happened. So this data is even more just like munged to the point where it's really hard to even explain what it is. So this is a way that we're quantifying, I guess, the population profile. So there's a profile of SNVs that corresponds to, you know, any particular time point. And so you can compare one point to another based on, you know, which SNVs were in the population. And so this shows the first decade on ordination. So basically what this says is that they're all pretty much the same with some variation, right? We know that from this, right? Some variation, but like the composition of the SNV profile is not that different compared to what I'm going to show you next. So 2011, 2012, which you could kind of see already from the plot that I showed, had something very strange going on where the composition of the single nucleotide variant profiles were shifting a lot. And then, you know, you get 2013, which is kind of still hanging out over on the right side of the plot, and 2014 might be coming back. Maybe 2015, 2019 is coming back, but we haven't sequenced more recently. So can't really tell you too much about post 2019. But if you zoom in on when that big change happened, I mean, I guess if you want to look right here, that I believe is August 3rd, 2012. There we go. And so you can see the abundance in those different time frames the diversity and the new SNVs that, would have appeared if it was some new population coming into the lake or completely emerging. I don't know how you would think about that. So we know that 2012 something strange happened, but we had never seen evidence that 2012 was particularly strange. Like we've looked at these data sets like every possible which way. And weird things that have happened is that in 2009, there was an invasive zooplankton. In 2015, there's an invasive zebra mussel, but this is like right in between those. So we didn't have any invasive species argument really to build around that. But Robin looked into the historical data, and this has a lot of different stuff on it. But really what it came down to is that it was a hot year and it was a dry year. So this shows the temperature as a function of the month for the years that we have sampled. And you can see that the red line is on the high end. It was also especially warm in the spring, which actually we found evidence, other data that like that spring warm spell seemed to have really altered what happened to the lake in the summer with other organisms, mainly the zooplankton. So yeah, you can see kind of the zooplankton mass was a lot higher during that year. Ice duration was lower than typical. And we had low overall abundance of phytoplankton, which would be cyanobacteria and algae, but that's probably because it was dry, right? There was just not a lot of rain that year. So something about, that aberration in the sort of, we call it the phenology of the lake and how it's sort of unfolded across 2012, we think is driving that. But do we have an explanation to report? No, like that's the best that we can do at this point. And that was enough to write a paper. So that we published this year. And I mean, I think one of the more interesting things that came out of that actually is that there are different populations that responded to whatever made that happen, like the dry, hot year, like whatever that did to the bacteria, in different ways. So these are now 3 different genomes within the phylum that the Nanopelagicus is in also, so the Actinomycetota. Some of them have like kind of a more gradual change in their diversity. Some have this step change that is more reminiscent of the one that I showed you earlier. And some changed, but then returned to their pre-disturbance levels. And again, like the interpretation of this y-axis is pretty brutal, so I'm not going to walk through it, but basically think of that as a measure of diversity in the population. So the point of that paper really was that there you can see signals of this ecosystem level change in the population structure of the bacteria, and they respond differently. I mean, depending on probably their phylogenetic placement, like we haven't actually looked that deep into it yet. So I want to give you some ideas to take home here, and then I think I have time to talk about the cyanobacteria too, which taking a similar approach, but this is all the published stuff from Robin. So we know we're able to see ecological dynamics. Like this is, microbiologists have been doing this now for whatever, 30 something years since 16S ribosomal RNA sequencing became a thing. And we know that things, increase in abundance and go down in abundance, and we're not exactly sure we can explain all of that, but now we can look at the population level and start asking questions about, where is that variation coming from? And does it have a consequence for the ecology? Why, what about that population level change? Is it gene content? Is it diversification in just some locus, like a handful of loci, or is it across the entire genome? So these are the kinds of questions that we're going to follow up on. That you have these really abundant cosmopolitan actinobacteria that are, have these different responses to whatever happened in 2012. Some of the environmental factors that we found can explain those dynamics, but we're really just getting started trying to unpack that, because for all, really, I was talking about 3 genomes, right? And remember that we have what is it, 2,700? And so this is probably, I mean, many of you may have the same problem that you generate this gigantic data set and you're like, whoa, look at all my data. And then you're like, I don't even know where to begin to analyze this, right? Because this is, I'm going to say, this is not hypothesis driven research. A lot of what we're doing is like looking at the data set and trying to like, okay, can we build a hypothesis around what happened in 2012? And then, maybe we can find a way to test that hypothesis. But it's like kind of the big data mining mindset that we're using here. So we could keep 20 more PhD students busy coming up with some of those ideas. So these are the just the top 50 genomes, and I wanted to show this as a so don't pay attention to this, but this is the range of nucleotide diversity that we have for those 50 genomes. And so the one that I was talking about a lot is way up here, right? So it actually it's very much on the high end of population level diversity. So I want to say just a few things about one on the other end of the spectrum, which is cyanobacteria. And this is the most abundant cyanobacteria in the lake. Across the entire data set. So, it blooms really intensely in June. And so if you just take, the fact that it's really like almost everything in the lake in June, it ends up being one of the most abundant, but it's also one of the least diverse in terms of its population. We have one genome from it, presumably, because there's only one species. But then you can see there's lots of variation based on what phylum they're in. So there's variation in the average, but then also in the variability of that across samples, which we'd love to try to figure out. So the cyanobacteria, though, I wanted to dig into just a little bit, and no need to worry about the y-axis. I need to tell my student that they need to figure out how to make those Professor Pet Peeve. I don't know if they're on the Zoom. Professor Pet Peeve. Make your axes with better labels. This is pretty fresh stuff. So we're showing nucleotide diversity with the cyanobacteria grouped based on their morphometry. Or morphology, because my student Chris has this idea that depending on how they grow, like in terms of their three-dimensional structure in the lake, that they have different ranges of population level diversity. So that organism that I mentioned that's really abundant, that has low diversity, is in this group right here. They form chains. They're also nitrogen-fixing cyanobacteria, which might be important. This is a colonial group called Microcystis. These are single cells that just they're kind of like Synechococcus or Prochlorococcus, if you're familiar with ocean cyanobacteria, and this is just another group. And these are another couple of very abundant groups. But this is the population that I'm going to expand a little bit more on. So this is also very fresh. Sorry for the horrible color. We hope Chris are you on there, Chris? It's fresh. They know that they need it. So this is an analysis that you can do with a new software package called Strong. If you folks do this, you could go dig it out and see if you can get it to work. It takes a lot of hacking to get it to work. But it deconvolutes the strains that are present in a sample. So remember, this is the most, or sort of the least diverse species. And this software package deconvoluted 5 haplotypes, or 6 haplotypes, right? Or if you want to think about them as strains. And these are samples which they had to be high enough coverage. So these were like days where it was blooming, right? So most of them are in July or late June or July. And here's one from November, but whatever. Also reappears in November. But I guess the take home message here is that, that's also variable, even though at the level of looking at nucleotide diversity, it's pretty consistent. So like now we're talking about even more entities that we have to keep track of. And, is it important that they're going up and down? Does it matter? Do they have different gene content? Do they have, functions that are different because they have variation in particular genes? So the little bit that we've been able to do digging into where some of this variation comes from, almost all of it that we found is phage defense. So things like restriction enzyme modification systems, changing the cell surface so that the viruses can't recognize them anymore. Toxin antitoxin systems, if you're familiar with that. So the rapid changes in diversity, right now we're hypothesizing are caused largely by phage attack or defense against phage, right? Rather than some kind of functional selection for their ability to take up nitrate or, something that is more ecological or environmental driver based rather than ecological interaction based predation. So this is something that we're following up on for those of you who know Jackie. Jackie, are you here, Jackie? I hope you have your mask on. Thank you. Is going to be following up on in her work. Let's see. Well, and that actually, I'm ending my story. I guess it's a little bit early, right? That's cool. More time for questions. I want to thank Robin. She persisted through her postdoc during the pandemic, and it was really painful. It also took her a long time to get through all the data, so it was a huge accomplishment for all of us to get that paper out earlier this year. Sarahi Garcia has been collaborating with us for a long time. Brett Baker was Robin's postdoc host. He's from Wisconsin, loves Lake Mendota. And so we went to him and said, hey, do you have a big computer? Do you want to help us with this? And he was like, because he wants to move here and be a professor here. But so if you're hiring a genomic scientist, let me know. And I'll tell him. So, and then Chris, who's about to graduate also, who worked a lot on cyanobacteria for their PhD. So also all the people that gave the funding, especially I'll give a big shout out to JGI. Some people really hate JGI for reasons I could get into, but they've been incredibly generous with us and super helpful. And like 90% of my research wouldn't be possible. That's a bit of an exaggeration, but 90% of this work wouldn't have been possible without them. So big thank you. All right. I would be happy to have questions. Thanks. Student first. Yeah.
It's fabulous talk and
Thanks.
to talk some more about all this cool data. I'm an ecologist, so of course I'm interested in the 2012 event.
Yes.
I'm curious from like a water quality perspective, if there's any kind of land use change, you kind of touched on that earlier, if there's any trends you see there in terms of something still or something that maybe selected for that, and then that population is then returned to evidence.
Robin, well, and I helped, looked at it like every possible which way to try to find, is there a smoking gun there? And the best that we came up with was the heat and low precipitation, which also so when we don't have rain, you don't have nutrients going into the lake. So then the phytoplankton populations, the algae and cyanobacteria are very low, right? So you had overall less primary productivity. So these bacteria that I've been talking about are heterotrophs. So they eat, we think they eat like things that get excreted by the phytoplankton. So there was one hypothesis would be that the food source changed or was much lower. And so maybe there was more competition there or something. And that's kind of the best that we've been able to come up with. There's not really any other signals that we have been able to. Agricultural, the nutrients are partly reflecting of that, but the agricultural, like the usual impact route is through rain, right? And when the rain falls, is the ground frozen and the manure, runs into the lake. So now I want to go back and look at the March precipitation. Because that might have mattered because it thawed so early, right? I didn't really think about that. It doesn't look like there was much going on there. Maybe a little bit, but certainly not like a usual year, right? That was the strange thing. Yeah, but it was like 80 degrees in March that year. It's very weird. Yeah. Good.
Right, I have so many things I'd love to talk about, but I'm going to limit myself to one question. So you sounds like you can now get haplotype, which means that you, in principle, can look at the combination and whether they're actually functioning like a population or they're functioning like a bunch of co-occurring eating presumably strains. To what degree in that spectrum they sit. That's something you can do with cyanobacteria, and could you ever do it with the Messier data set?
Well, the dirty secret here is always the short reads. So when you assemble a mag, it truly could be a a population genome. So you can't really say for certain that the MAG is a true entity. So Jackie's looking a little bit at that because we also have single cell genomes. And for the most part, the MAGs actually look really good. They look like the single cell genomes, in terms of gene content and other factors. What was I gonna say? But you don't get linkage with the reads, right? Because they're too short.
It's cyanobacteria.
That is a very fancy workflow that I'm not sure I could explain how it works, but it is able to use assembly graphs. So it uses the assemblies to deconvolute the strains, and it only uses really conserved marker genes rather than the entire genome, which is also something that is kind of a caveat, right? There's probably even more variation because it's only looking at the really, really conserved ones. There's a couple of new software packages that have just come out in the last month that we might look at also that seem to be able to handle more genes. But we're also looking at long read sequencing. I don't think that, I'm always skeptical of the short reads for doing that, no matter how good the software is, right? But, if we can get long reads with PacBio or MinION or something, then you can start looking at recombination. You have to have enough coverage, right? So one struggle we have with these is getting high quality long read or long high molecular weight DNA. So that's a challenge too, but it's getting close. We're getting closer. And I mean, we might abandon Illumina altogether once we can get that going for exactly that reason. Yeah.
There's a question on Zoom
Oh.
from Aaron.
Uh-huh.
Thinking of the variation in pi over time, how sensitive is the measurement to sampling strategy? Meaning, will differences in location or clustering of samples at different time points affect the observed variation? I don't have a good sense of how well mixed Lake Mendota is, and if there is something you have to think hard about in collecting time series data.
Yeah, we talk about this all the time. I mean, you can't be everywhere all the time and collecting samples, right? So you have to decide, and the LTER decided a long time ago to do it in the middle, like the deepest part of the lake. And so that's where all this data comes from, is one point in the lake. But we know that there's spatial heterogeneity across the lake. You can see it from space, right? And so that's one of Chris's chapters, is to take transects out from the shoreline to look at differences in cyanobacterial population, or more like species composition, community composition. And they're finding that there's a lot of nitrogen fixing bacteria that seem to like to be around the edges, which might explain why the blooms seem to form there and then move around the lake. But we don't have good enough metagenomes from those samples. So I'm not sure we're going to get to that question about strain level variation, but we definitely see species level differences. Yeah. Yeah.
So you talked a lot about the drought year 2012, which obviously is the big outlier. But I have a personal interest in the flood year.
Yes.
2018.
Did your house flood?
So I mean, it was once in a millennium. Pre-climate change. So is there anything that stands out about 2018? That would have discharged an enormous amount of water
and nutrients
not just
coming
phosphorus
with
from
it.
the farm, but I mean the city, right? Dumping all kinds of, garbage from the streets.
Yep. And overflowing, well, sewers that may have overflowed a bit. I mean, part of the problem is that our 2018 is, in this particular species, I'm not sure that you could say there's any signal. Water quality wise, like just the abundance of the cyanobacteria. I'm trying to remember if we've looked at that. I can't say that we've seen a lot of signal. There is a new cyanobacteria that has colonized the lake that appeared in 2017. But that wouldn't be the flood. So no, we don't see a lot of it. But we might not be looking at the right thing, right? Maybe there's some other variable in the lake. I mean, but 2008 was another crazy flood year. Was that before your time here? No, you've been here almost as long as me. 2008 was like epic. Okay. Yeah. So that I was here. I didn't realize you were so much younger than me. 2008 was almost it maybe worse in some ways in terms of the intensity of precipitation that came all in a very short amount of time. And we can see some signals from that in other metrics, but yeah, maybe not. I don't know. It's hard to say. Is that something? Yeah, so you can look at it all kinds of different ways, and sometimes you see a thing, other times not. Yeah. Yeah, that's okay.
Great talk. I'm just really curious, that high plot, we see that explosion in 2012, and it doesn't really seem to come down.
Right.
Versus when you had the MES plot, you see it kind of returning back. So what's that, what's?
I'm trying to remember if that ended up being in the, if we had an explanation for that in the paper. Let me go back here. So 2012, it definitely didn't come back. It's like gradually coming back in terms of the SNV profile. So the SNV profile includes both the locus and the frequency, I think. Yeah, no, maybe it's just the locus. I think the SNV profile here is just, was there a variant at that position, but doesn't capture the number of reads that had that amount of variation. That would, we'd have to calculate that in a different way. And it's really hard to interpret. So it could be that the positions weren't changing. The positions were changing, but the level of variation at each position was changing. If that makes any sense? It starts to really bake your brain. And yeah. Although,
I think something's maintaining that, right?
Yeah. Yeah. Right. So what the ordination is based on is counting the columns that have differences, right? And so how many columns have a difference on that date versus not? But I don't think that it captures that the fact that, like, maybe, can you see that green one? But like, part of the, or maybe some of these are better, right? So like part of the population doesn't have it and the other part doesn't. And I don't think that that metric captures if it's split 50-50, whereas the pie does. Wait, does pie capture it? Yeah. Yeah, it does. So they're measuring different things, which is another challenge. And this is all, this is another confounding thing, is that this is all with respect to a reference genome. Right. It was the one that was most complete, but there were probably 470 versions of that genome that were in the data set that we chose not to use. Yeah.
It seems like you picked name of planet because it was a cosmopolitan abundance of disease. And then you found that they had an odd diversity pattern in 2012, which that's cool and interesting. And then just wondering, like, sort of in reference to Chris's question, can you sort of reverse the process and say, given something weird happened in 2018, can I identify which genomes showed a weird pattern due to that event? What species arose became abundant in that particular year?
Yeah, and we've done that with 16S, so at more the kind of the community composition level, because we knew that, well, we didn't know going in necessarily, but we figured it out pretty fast, because there was these two invasive species, and so the community composition responded really strongly to that, mostly in the cyanobacteria, actually. So we have a paper where we talk about how the blooms of the aphanizomenon, that one that I dug into with the 6 haplotypes, is coming kind of earlier in the year. I don't know if people pay attention, but we've had a couple of blooms in May, which is very unusual. And so that seems to have come from these 2 invasive species, or was coincident with, right? So and we also are looking at other things in the silica concentration, like, go figure, like, happens to be some years really high and some years really low. And we know that has to do with diatoms that are in the lake, but that seems to be causing differences in the bacteria, which is probably some food web thing, right? There's like things that eat the diatoms, and then there's things that some of the diatoms are heterotrophic. Are they heterotrophic? There are some things that are heterotrophic that get eaten and they might eat the bacteria, but they go away. And so, right. So you start unraveling it and it gets complicated fast, obviously. But we went for the low hanging fruit, obviously, right? For the first paper, which was the 2012 emergent signal. But yeah, we're doubling down on the invasive species because we see lots of cool stuff because of those. Yes, Keely.
Maybe Chris would be my first ask, but looking at the advanced nominal strain graph that they made, do they have an inkling as to why some of the haplotypes, like the zero, you can see it was a lot more intense, like back in 2004, and then kind of gradually been less intense. Is there a reason why?
Oh, we have no idea yet. We have no idea yet. And part of the limitation of this technique is that it gives us a collection of marker genes that's fairly constrained. It's what, like, Jackie, do you know, is it like 20 or something? I'm not even sure, but it's not the whole genome, right? So there's things that we can't easily link between these 6 haplotypes and what we're seeing at the whole genome level. And that's kind of one of the things that I think Jackie is going to be working on trying to tease out, because if we could link these to the rest of the genome, we would have a better answer to that. But right now, they're different. Microbiologists who study, environmental microbiologists spend a lot of time going, oh, it's different. And scientifically, that's where we're at. Yeah.
I don't know if you mentioned this, so sorry, but do you know anything about how spatially variable species compositions are, and how if there is a lot of variation that affects it, how you pick where it's happening from?
Yeah, I mean, it's also so complicated. So, like, was it windy yesterday, right? If it was windy, actually, you couldn't go sampling because that's dangerous. So, like, there's that too, right? But wind should mix it. And we can see that, right? You have waves. Was it really calm for the last week? In which case, there may be a lot of heterogeneity. And there's lots of different things that you can measure. Obviously, there's the bacteria, but you can measure things like, chlorophyll, which is a signal for the algae and the cyanobacteria. You can measure, sometimes conductivity, like salt, like there's a surprising amount of variation in the XY plane. There's also variation in the Z, but it's hard to study because you have to be everywhere all at once. So our colleagues have these boats that are equipped with a sampling thing that like sucks the water in continuously and puts it past sensors to measure things like temperature varies a lot. Temperature, chlorophyll, methane. So they want to see where there's hotspots of methane. And they just drive around the lake and collect data, but you still just get a track around it. You can't be in every single location. But so Chris actually teamed up with them to look at some of the spatial variation in the cyanobacteria, but those data are still being processed. We know they're different. How different? Does it matter? That's the next question. Thank you.
Right,